Okay, so welcome to our presentation. We're going to talk to you about Drosophila melanogaster and the mutations we studied. My name is Carly Duval.
I'm Felicia D'Alessandra.
And here we go. So, as we talk today, I'd like you to think about how it's possible if we only worked with 2 mutant phenotypes and 1 normal phenotype, how is possible that we got 4 phenotypes at the end, which is shown on the picture here. You can see we have one with white eyes, one with scarlet eyes, and one with wild type eyes, and then this weird brown color.
So this investigation was possible in the time period of 10 weeks because of the subjects of study. Fruit flies or Drosophila melanogaster are perfect model organisms because of their fast life cycle, which is completed in 10 to 15 days. They require low maintenance, they have a small size and simplicity. They are simple organisms in regards to their genetic information because they have only 4 pairs of chromosomes, which allows us to observe and become proficient with the basic studies of genetics.
And as you can see in the picture here on the side, we have the male and the female adults that breed and create the embryo, and then they go through this life cycle here, all the way to pupa and then back to adult. And then, so for male and female flies, throughout our experiment, we had to determine the sex of male and female flies and split them by gender in order to get successful crosses of the different, in order to split them by specific sex, in order to get successful crosses of the different mutations we studied. As you can see in this picture, we have a male on the left and a female on the right. The males have a black and round abdomen. They're also smaller than the females, and they have sex combs, which you can see on the image. They're little black dots on their front legs. The females have pointed and striped abdomens and are often larger than the male fruit flies. The easiest way to tell the difference between them is by looking at their abdomens because they are so different.
Okay, so both of our mutants that we will talk about in a second affected the eye color of the flies through disrupting pigment presentation. The wild type, which is the normal eye color, comes from the presentation of 2 pigments. One is called the omicron pigment, which is the rusty color shown in the picture on the top, while the second pigment is called the pteridine, which is the bright red, which is almost a Coca-Cola red color, which is shown in the picture on the bottom. When these 2 pigments are combined, it causes our wild type phenotype. The flies that have a scarlet phenotype, who we will talk about in a second, are the single mutant flies that we observed. The scarlet mutation is caused by the prevention of the transfer of the pigment omicron, which is what causes the rusty color. This leaves only the bright red color being transferred to the eye of the fly, which results in our scarlet mutation, which you can see in the presentation. The brown mutation that we studied was our double mutant. This means that the fly studied had both the scarlet and the brown mutations. The brown mutation specifically is a lot like the scarlet because it also disrupts a transporter, but this mutation disrupts the transporter that imports the guanine, which is essential to have the pteridine pigment. Now that you know a little bit about the colors of the eyes and how they can be influenced, we can now look at the specific mutants that we studied.
So this is our wild type phenotype. It's one of the 4 phenotypes we observed over our experiment. They're most easily identified by the color of their eye, as well as the visible cytopupil, which can be seen in the picture. The cytopupil can be seen as the dot that's slightly darker than the rest of the eye. It's kind of within the eye. The wild type variety is the visual representation of the 2 pigments being expressed as a combined phenotype, and is what we call the wild type phenotype.
The scarlet phenotype is one of the 4 phenotypes that we observed over our experiment, and they were also our single mutant flies. They can be recognized by the eye color, as their eye color was much brighter than the wild type. The scarlet phenotype is one which we have described as a Coca-Cola red. They have no visible cytopupil. For the molecular information for this mutation, we found it on Flybase. To begin with the gene, it is labeled as ST, which being lowercase shows that the gene is recessive. And for the cytogenetic location, it is placed on the third chromosome on the left arm, which translates to the third chromosome on the left arm. And the mutation occurs from the nucleotide 16,497,000 to 651 to the nucleotide 16,500,460, which translates to about 2,810 nucleotides long and is on the plus DNA strand. The flies that have a scarlet phenotype have a mutation that prevents the production of a transporter protein that transfers the pigment omicron. This is the pigment which causes the rusty color. So in the absence of that, only the bright red color is being transferred to the eye, which results in the scarlet mutation, which you can see in the pictures on the slide. There has not been any recorded cases of humans having this mutation, but other model organisms like mosquitoes or roundworms also have these mutations.
So for another mutation we studied, this is the scarlet brown phenotype, which is one of the 4 phenotypes we observed, and also are double mutants. They can be recognized by the white color of their eyes and that they don't have a cytopupil. We found that the molecular information for this mutation on Flybase, to begin with it, the gene is labeled as BW, which being lowercase shows that the gene is recessive, like the scarlet gene. For the cytogenetic location, it's placed on 2R, which translates to the second chromosome on the right arm, and the mutation occurs from the nucleotide 25,527,805 to the nucleotide 25,538,499, which translates to about 10,694 nucleotides long and is on the negative DNA strand. The brown mutation is a lot like the scarlet because it also disrupts the transporters, but this mutation disrupts the transportators that import guanine, which is essential to have the tyrosine pigment. And it also affects the tyrosine pigment and not the omicron pigment. Humans do not have this mutation, but mosquitoes do. However, it's important to realize that these double mutants on the slide have the brown mutation, but they also have the scarlet mutation. So this isn't the visual representation of the brown mutation. It's just kind of what we studied that we knew had the brown mutation. This combination creates the white-eyed fly because it has the scarlet mutation and the brown mutation and disrupts both pigments of the eye.
So the Punnett squares on this slide shows the single mutant parental cross, which resulted in the F1 generation, which were then bred into the F1 cross to create our F2 generation. The first Punnett square shows the breeding of the parental stalks to get the F1 generation. As you can see, all of the offspring from that cross should be wild type, but they should be heterozygotes, meaning that they all carry both of the genes. The second cross, the breeding of the offspring from the first generation, is shown by the second Punnett square. All of the offspring should be heterozygotes, meaning if the genes follow Mendelian genetics, there should be 1 homozygous wild-type fly and 2 heterozygotes who still have the wild-type phenotype, while there should be 1 fly out of the 4, which is a homozygous for the recessive scarlet mutation, and will have that phenotype.
So these Punnett squares are for the double mutants, and they show the parental cross right here in the table with all the blue, which resulted from the F1 generation, or the blue boxes are the F1 generation, and they were bred to create the F2 generation, which is shown by the second table. But the F1 cross was bred to create the F2 generation. The first Punnett square shows the breeding of the parental stalks to get the F1 generation. As you can see, all of the offspring from that cross should have a wild type phenotype, but they should be heterozygotes, meaning that they're carrying both genes. The second cross, the breeding of the offspring from the first generation, is shown by the second Punnett square. All of the offspring should be heterozygotes, meaning if the genes follow Mendelian genetics, there should be one homogeneous wild type fly, and right here, shown right here, and 6 heterozygotes who still have the wild type phenotype. So the rest of these blue boxes here, there should be 3 that have the rust phenotype, as they are homozygous for the recessive BW gene and have at least 1 wild type scarlet gene. The same is true for the scarlet color. There should be 1 fly out of the 4, which is a homozygous for both the recessive scarlet and the recessive brown. And this is what has the white phenotype. And we will analyze the data through gel electrophoresis and chi-square analysis.
Okay, in our experiment, gel electrophoresis process was used to separate the different size DNA fragments in order to determine if the correct fragments were present in the organisms that we studied. Both the F1 and F2 generations were used to determine if there were heterozygotes in the crosses. Based on our results, we were able to conclude whether the crosses of the single and double mutant flies were effective.
So for the single mutant results, we predicted that the F1 generation will appear to be all wild type flies with a 50-50 ratio between the males and the females. We decided this because the wild type phenotype would be dominant over the scarlet mutation, and the mutation is not sex linked. So on this table here, you can see we have the I phenotype, and because it's only the single mutant, we just have wild type and scarlet. And then those are separated into male and female. And then these numbers here are what we counted. There's in the cross with the wild type fly, crossed with the single mutant fly, and we got 29 male wild type phenotypes and 54 female wild type phenotypes. In the second cross, we did scarlet phenotype crossed with wild type phenotype, and we had 40 wild type male phenotypes and 44 females with the wild type phenotypes, which brings us to a total of 69 wild type male phenotypes and 98 wild type females.
So for our molecular results, we used the E. coli and the Puvil primers for our single mutant PCR test. We expected to see bands that show the fragments for the scarlet DNA as shown in our drawing of our expected results. Unfortunately, we did not see any bands on our gel electrophoresis, so we were unable to confirm or deny the results confirming our hypothesis.
So on this table, on the slide, it shows the results of the 2 phenotypes. The observed value is the number of the flies counted for each phenotype. The expected value was calculated by multiplying the expected ratio by the total number of flies. Then the chi-square analysis value was calculated from the formula found underneath the table. The next step was to take the 7.01 number, which we got by adding the 2 chi-square analysis numbers together, and look at the chart in lab 5 to find how the number stacks up with a degree of freedom of 1. The p-value is larger than 0.05, so it's found that our data does not support the expected ratio. For the possible errors, we think we probably miscounted some of the flies. The number of wild type flies is still much larger than the number of scarlet flies, so we still determined that the scarlet mutation is an autosomal recessive trait.
So for our double mutants, we predicted that the F1 generation will appear to show all wild type phenotypes with the 50-50 ratio between male and females. This would prove that the wild type phenotype would be dominant over the mutation of the scarlet, brown, and white phenotype, and that it is not sex linked. So in the table, we showed that we studied the wild type, scarlet, brown, and white phenotypes. And in the first cross, we see a scarlet brown female crossed with a wild type female. And it's shown that we were resulted with 29 male wild type flies and 54 female wild type flies. And then for our second cross, it was just the reciprocal of that, which were scarlet brown males crossed with wild type females. And that resulted in 40 male wild type flies and 44 female wild type flies. And as the table shows, you can see that we were only given wild type flies and the scarlet, brown, and white phenotypes were not present.
So for the double mutant PCR, we used the 412 up and the 412 down primers, along with the same ladders we used in the single mutant PCR test. We expected that the wild type and the scarlet gene fragments would be visible. Through the picture of the gel electrophoresis results, we see that these results matched our hypothesis, excluding the second column, for which we received no results for. So this orange in the lane 2, you can see we didn't get any results for that lane, but the other 3 did match our predictions.
The table on this slide shows the results of the 4 different phenotypes present in the cross. The observed value is the number of flies counted for each phenotype. The expected value was calculated by multiplying the expected ratio by the total number of flies. Then we use this chi-square value was calculated by the formula found underneath the table. The value 25.13 was taken and compared with the table from Lab 5 by using the degree of freedom of 4 to determine how our calculated value stacks up with those given values under that degree on the table. Considering that our p-value is larger than 0.05, we concluded that our data does not support the expected ratio that we predicted, and we determined that there was no epistasis effect due to the fact that we were visibly able to see all 4 eye colors. Therefore, we decided that possible errors that took place were not realizing that there was an actual 4th color present, and in general, probably just miscounting the flies based on their phenotypes. The number of wild type flies that were observed are significantly larger than the scarlet, brown, or white mutations that we observed. So therefore, we concluded that these mutations are also autosomal recessive traits.
So the 2 pictures on this slide represent the genetic location of where the scarlet gene and the brown gene mutations take place. The yellow region on each of the pictures highlights where this happens. The scarlet gene is located on the 3rd chromosome on the left arm, and the brown gene is located on the 2nd chromosome on the right arm.
So we decided that if we were to do a study or experiment like this in the future, we would look into other mutations in flies as they are easy to work with and have a fast life cycle. We could look into human genome, even though it's way bigger and more complex. We now know a lot more of the basics, and that could allow us to kind of understand the parts of it differently. We would also be able to look into other genetic mutations, maybe diseases, inherited traits that make us so diverse, selective breeding, and how it can affect things, or even genetics over time. All of these topics are things that we were interested to learn more about, as we learned about them on the surface level this semester, but we feel as though we could start diving deeper into the topics themselves.
So we recommend students who are taking this class start as early as you can with each of the crosses and take your time counting the flies. We both learned personally how to care for, examine, and successfully set up stocks of flies, how to perform a successful gel electrophoresis, do a PCR, and use chi-square analysis to see how close expected and observed values are, and kind of think about what we could have done wrong or why our results wouldn't match up. Thank you for listening to our presentation. Hope you enjoyed.